right. Good morning, everybody. So now let's finish the first part of a fuel utilization in response to food intake. And I think we discussed about the different tissues utilize different metabolic fuels in different physiological conditions, right? And I finished brain, liver, small intestine, large intestine. Now I guess we stopped here, right? So now adipose tissue. So adipose tissue, the primary fuel that adipose tissue can use glucose and triglyceride, like carbohydrate and fat, and in different physiological conditions, adipose tissue can take up, metabolic fuels and in different purpose. For example, in a fat condition, adipose tissue can take up glucose, of course, for oxidize, right, for energy and also for, but in particularly in adipose tissue, also for converted glucose to glycerol 3-phosphate for triglyceride synthesis, right, as a glycerol backbone. And fatty acids in the fat condition, adipose tissue pick up lots of fatty acids, right? The purpose of picking up fatty acids is for synthesis of triglyceride and stored in the adipose tissue, right? Because adipose tissue is a primary site for fat storage. So a lot of fatty acids flow into the adipose tissue and stored in the form of triglyceride. But during the fasting condition, and because adipose tissue also is a major provider of fatty acids, right? So fatty acids will release, a lot of fatty acids will be released from adipose tissue in the fasting condition into the circulation and utilized by other tissues, and also meantime release glycerol, right? Basically it's, from the hydrolysis of triglyceride in adipose tissue. And lactate can also be released from adipose tissue, but not like a lot, because glucose will be partially oxidized in the adipose tissue during the fasting. Again, I mentioned a lot, right? During the fasting condition, most of tissues partially oxidize the glucose. So lactate will be also released from adipose tissue. Okay, so and in just give a little bit more information during the fat condition, we said the adipose tissue pick up a lot of fatty acids from circulation, right? And which enzyme you guys remember that hydrolyze triglyceride in the two important lipoproteins, chylomicron and VLDL. Lipoprotein lipase, right? LPL, and that usually synthesized and secreted during the fat condition by insulin stimulation, and this LPS, main function of LPS is to hydrolyze triglyceride in VLD and chylomicron, so then release fatty acids then can be picked up by adipose tissue. So and in the fasting condition, again in adipose tissue also triglyceride need to be hydrolyzed and release fatty acid into circulation, and the key enzyme for lipolysis or hydrolysis of triglyceride in adipose tissue is hormone-sensitive lipase, right? We did a comparison between these two, lipase in different physiological conditions, and hydrolyze different substrate. Okay, now we start to talk about the adipose tissue function in fuel metabolism. We mentioned that adipose tissue has a very important secretory function, right? Can secrete very interesting hormones and regulates whole body metabolism. So we're going to talk about this. And before talk about the adipose tissue function, I just want to go over again the three nutrients interconversion. Remember that we know that protein is able to be used to make glucose, right, through the gluconeogenic pathways. And carbohydrate, and both carbohydrate and protein can also be used for make fat during the fat condition when we have too much glucose or too much protein or amino acids from the diet, and this can be converted to fat and stored in the adipose tissue for later use during the fasting condition. But the question, we also know answers, right? The fat cannot be converted back to carbohydrate or protein, right? Once they, converted from carbohydrate protein and deposited in adipose tissue. So what are we going to do to get rid of fat if we don't need that much fat? We have to have a efficient way, right, to get rid of fat and this kind of, the regulating mechanism. So how our body will do to remove or to reduce the fat. What's that? Yeah, but if we have too much, how we wanted to reduce. Is that? Burn it. Yes. Excellent. Burn it, right? Burn fat. So which metabolic pathway do you think is the efficient way to burn fat? Which metabolic pathway? Oxidation, right? We oxidize fat. So fatty acid oxidation or beta oxidation is a very important, most important pathway that can get rid of fat, right? We burn, fatty acids and break down fatty acids, triglyceride fatty acids, and burn, oxidize, and produce energy. And of course, there are other mechanisms. When we utilize fat or fatty acid, oxidized fatty acids, we're not only just to make ATPs, but our body also be able to convert the energy to heat, right? So then so this is a very important pathway called energy expenditure, right? We use the energy that can help our body to reduce the fat. So, and obesity and metabolic syndrome, I guess you guys heard a lot about this, right? But in terms of mechanism, in general, obesity developed is due to the imbalance between energy intake and energy expenditure, right? So any pathway, either food intake or energy expenditure, is impaired or is imbalanced, that can cause the obesity. For example, if we eat too much and our body cannot cannot use actual energies, or the other way that if we have some problems with the energy expenditure, then this will all causing the obesity, right? And so very unfortunately, the United States also leading the prevalence of obesity. And I think this was probably many years ago, and now I think the rate of obesity is about like 33%, right? So it's really high. And I think the government, like NIH, invests a lot of money to study obesity, try to prevent or reduce the obesity rate. But I don't know, but it seems like the rate still remain high. So I don't know, maybe we need to thinking about the research approach that is whether it's effective or whatever. So this is just the obesity rate map. Just give you some idea. You know, in 1990, that a lot of American people are kind of lean, skin, right? But now it's a lot of increase in obesity rate in the whole United States, but not in the United States. Actually, also globally, right? The obesity become like epidemic. So body mass index is a way to measure the or to define the obesity. Body mass index is the weight divided by the height square. So body mass index, we wish we will have a desirable range, which is between 20 and 25. And if the BMI is between 25 and 30, is considered overweight, and 30 to 40 is considered obesity, and greater than 40 is severe obesity, right? So and the body composition, what percentage of a protein, fat, and the water and protein and minerals. Actually it's gender difference. Male or female, they have a different body compositions. For example, male usually have a little bit higher percentage of water and than female. And females have a little bit higher, percentage of fat than the male. Yeah. And fat distribution is very important. We know obesity is due to, in general, is due to you have too much fat accumulating in the body, but the fat distribution in which region, in which depot is more important in terms of the the relationship between obesity and metabolic syndrome. So obesity can be categorized to abdominal obesity and hip side obesity. So abdominal obesity means that the fat mostly accumulates in the abdominal region, right? And this type of obesity is more common in men. So sometimes people also call the man pattern obesity, like apple obesity. And the hip side obesity is considered a female pattern of obesity. That means the fat mostly accumulated in the hip side region, the subacute, under skin region, but not much in the cavity, abdominal cavity. So the female is also is kind of interesting. The female before the menopausal age, the female tend to protect from the obesity and metabolic syndrome because of estrogen level pretty high. But after the postmenopausal, the woman seems to be an increased risk for development of obesity and metabolic syndrome because of estrogen level it goes down. And also the fat distribution is changed. For example, before postmenopausal, the most of the fat actually accumulate in the subacute region, right? And after postmenopausal, we start accumulating in the abdominal. So the switch of fat distribution with the age. So the region of adipose tissue or fat tissue accumulation is very important and very close correlated with the metabolic syndrome. So and other the thing is that the up body fat distribution, also visual obesity, just similar to what I described in apple obesity or male pattern obesity. This visual obesity more closely related with the metabolic syndrome. means that visceral obesity have a high risk for the development of obesity and development of metabolic syndrome. And so there is a way that we can predict the risk for the obesity called waist to hip ratio. We measure the weight hip ratio, and it basically is a circumference of a waist and versus a circumference hips, and this ratio can help us to predict the risk, right, of the metabolic disease, metabolic complications. For example, if the ratio is greater than 0.85 for men and or 0.95 for 0.85 for women and or 0.895 for men, then if ratio above or greater than this this number, then the man or woman will have a higher risk for the metabolic syndrome development, right? And other concept that I wanted to bring in is called epitopic fat deposition. We know that adipose tissue should be the right place, right, for storage of fat. So if somehow the adipose tissue function is abnormal and cannot accumulate, cannot store fat, so the fat will go to the other non-adipose tissue, for example, liver, heart, and muscle can also accumulate the fat tissue in the pathologic condition, right? So the epitopic fat deposition means that fat accumulates in the non-adipose tissue in the pathologic conditions. And function of white adipose tissue. Of course, we know that the store fat, right? Store triglyceride is the main function of white adipose tissue. And also the fat buffering system in the lipid homeostasis means that we wanted to maintain the normal levels of lipids, triglycerides, fatty acids in the circulation or in other tissues. So the fat tissue is very important, serves as the buffering system, right? You can either to store the excess fat in triglyceride in the adipose tissue, so allow the less free fatty acids being around in the circulation. So it certainly is sort of a buffering system. And the second important function is glucose homeostasis, right? Fat is or tissue is also important that pick up glucose in response to insulin. Remember, the GLUT4 expression level is quite high in adipose tissue, although it's relatively small compared to muscle. So remember, the muscle takes up 80% of insulin-stimulated glucose uptake, and versus 20% of insulin-stimulated glucose uptake occurs in the adipose tissue. But adipose tissue does take up glucose, right? So it is also contributed to maintaining the blood glucose level, but not just this. Take up glucose can directly regulate glucose level, but adipose tissue is also secretes some very important hormone that we're going to talk about later. This hormone can also regulate whole body metabolism, right, the glucose uptake. And then lastly, very important that function of adipose tissue is endocrine function. So I think since 1994 was when the first adipose tissue-derived hormones was discovered, the leptin. Since then, the people totally changed, the concept that the adipose tissue is also very active organ that can involve, metabolism by secreting important hormones and peptides, right? So, and regulates whole body energy metabolism. Now let's take a look at the adipocyte. How adipocyte look like, right? This is just a cartoon show you the adipocyte. So adipocyte is morphologically very different from other type of cells. And it's larger, right? And most importantly, these are unilateral lipid droplets, right? Filled with the lipid droplets. Adipocyte have very small cytoplasma, and the nucleus actually, pushed by the lipids to the edge of the cells. So this is a cartoon show how fat cell look like, but I'm going to show you the real fat cells, right? And so my group actually research work on the adipocyte, adipose tissue function. So this is something that I did maybe more than 10 years ago. And I isolated fat cells from rat adipose tissue and look at them under a microscope, so you can see this. This is not a lipid droplet. I mean, the oil drop is actually real fat cells, right? So you see the fat cell, really looks like the blue bubbles, and you don't see clearly nuclear, right? Very difficult to see, but this all filled with lipids, triglyceride oil. Right? And adipocyte has insulin receptors, right? So it's responsive to insulin, and also have a the adrenaline receptor. Adrenaline receptor responds to catecholamine, epinephrine, norepinephrine. So, and we also know that the adipocyte can synthesize and secrete the LPL, right? And LPL, the main function is to hydrolyze triglyceride in chylomicron VLDL and release fatty acids in the fat condition. And fatty acids are picked up by fat cells and resynthesize triglycerides stored in the fat cell, right? Remember this process. We talk a lot. And also in the fat conditions that glucose can be picked up by adipocyte and in response to insulin, right? Insulin receptors. So GLUT4 is the main transporters that pick up glucose into the adipocyte. And this glucose can be utilized for synthesize the fatty acids and also glycerol 3 phosphate backbone for triglyceride synthesis, right? Remember that. And during the fasting conditions, because of the catecholamine levels increase, so catecholamine can bind the receptor on the adipocyte and stimulate the HSL, hormone-sensitive lipase, right? So hormone-sensitive lipase can hydrolyze triglyceride and release fatty acids of glycerol. And NEFR is the, I think it's free non-esterified fatty acids, right? So non-esterified fatty acids, also called, sometimes we just give a different name, right? So free fatty acids or non-esterified fatty acids. All right, so now secretory function, right? The last slide we just talked the basic function, store fat and release fat during fat fasting condition and provide energy for body use. And besides that, the adipocyte also have an important secretory function, endocrine function, and can release a lot of things. Here, the first we know, of course, release fatty acids, right, during fasting and provide energy fuel for other tissue. And very important hormones like leptin. Have you guys heard about leptin before? Good. So leptin is a very important hormone that's secreted by adipocyte, and also a lot of other cytokines like TNF alpha and IL-6, or other cytokines. So adipocyte sometimes also function as the immune cells, right? Secrete a lot of cytokines and involve the regulation of immune functions. And the angiogenesis, angiotensinogen, right? These factors can regulate the blood pressure, right? The cardiovascular functions. And adiponectin is the other important secreted factor hormones that can regulate energy metabolism. So we're going to talk about these two, leptin and adiponectin, how this adipocyte-derived hormones regulate energy metabolism, right? And resistin is another one that can also secrete from fat cells and regulate some immune function. But compared to leptin, adiponectin, and this one, yeah, the question?
I guess what we need to know about all of these, we just need to memorize what they secrete, or?
No, I think it's just the concept, the fat cell can secrete it, this kind of things, and we just wanted to focus on this too. So you guys probably wanted to remember these two hormones, how they regulate the metabolism, right? And just general concept, adipocyte, not just a store and release fatty acids, but also important secreted peptides, right? Regulates function, and you guys probably wanted to focus on these two hormones, right? Okay, so now leptin. So let's talk about leptin. So leptin, the secreted from fat tissues into the circulation, and leptin has two targets. One is called central target in the brain, right? And then the other target, the peripheral target, particularly in the skeletal muscle. So leptin released from fat cells and go to the brain and binds the receptor in the particular region. Mostly the target of leptin in the brain, in the hypothalamus region. But you probably don't need to remember this, I guess. But just the specific neuron that can be bound by leptin and activated by leptin. And so one of the function is called central function, is bind the hypothalamus neuron to suppress the food intake, appetite. So leptin can inhibit the food intake, right? So when we have a high leptin levels and our appetite goes down, right? We don't want to eat. And this is one important function, right? Remember the balance, energy metabolism, energy intake, energy expenditure, the very important balance, right? So food intake is one of the aspect of the energy balance. So once we inhibit food intake and we eat less, right, you can manage the body weight. And the second target of leptin is to stimulate fat oxidation in the skeletal muscle. So skeletal muscle is the second target of the leptin. So leptin can also goes to the skeletal muscle and binds receptor on the surface of the muscle fibers and can stimulate the fatty acids oxidation, right? So increase the energy expenditure. So the net effect of the leptin will be good, right? If we have a high levels leptin, then we'll have a reduced food intake, eat less, and burn more fat, right? So increase energy expenditure. All right. Any questions about leptin? Right. So, and how leptin is regulated, that is also related to the feeding and fasting, right? Because it's regular energy metabolism. So if we fast animals for 24 hours, we can cause the marked reduction in the leptin gene expression, like 80% reduction, which makes sense, right? During the fasting, we don't want to burn fat, right? We want to save energy. So during fasting, we want to have our leptin levels keep low. So fasting causing the decreased leptin expression, and refeeding when we refeed animal, and then can rapidly reinduce leptin gene expression, right? To do 3 to 6 hours, and you see leptin levels start to increase, and then within the 24 hours, leptin levels return to a normal level. I mean, the fat condition. Right? And so people also thinking maybe fasting fat condition, the regular leptin is mediated by insulin. So because insulin levels change, right? Always, fasting condition. So what they did study, and they first of all they fast animal. So then we expect insulin level will be low, and also leptin level low, right? So then they give insulin to the fasted animal and see if insulin can raise the leptin expression, raise leptin level, and actually yes, and it does. So insulin is given to the fasted mice can increase the leptin expression. So from this experiment, they make a conclusion that insulin actually is a hormone that does regulate the leptin levels, right? So here's a question: why does the body make less leptin during fasting? I already told you, right? Answer. So we don't want to burn too much, right? Increase energy expenditure during fasting. We want to save energy during the fasting, so we need to make leptin levels low. All right. So kind of interesting stuff, I guess. So OB mice, the leptin deficiency. So OB mice is a mutant mice. It was discovered like a long time ago, 1949, at the Jackson Laboratory, and it's just the E. colony somehow mutated, and then this mutant mice, and when they were born, they actually you cannot see the much difference in between normal and OB mice, but when they grow up and they actually develop very severe obesity and also type 2 diabetes, they have a very high levels of blood glucose, and they also eat a lot compared to the normal mice. So what gene is mutated in the OB mice it was not very clear until 1994. A group of researchers, and I think at the Rockefeller University, and they actually cloned the leptin, the gene that they found that actually is the leptin, that mutation that caused the problem in the OB mice. So the other experiment is also kind of interesting. They call it parabiosis, the cross circulation between normal and OB mice, and they found that OB mice, once they received kind of circulation from the lean or normal mice, and they actually the obese phenotype actually reduced. So they believe that some circulating factors that goes from normal mice to the obese mice then cure the obesity. So and until 1994 that they discovered that leptin actually is a gene that is mutated in obese mice that causes all the problems. So and they also inject the leptin back into the obese mice can also reduce the body weight and reduce the other diabetic syndrome or problems. So which makes sense, right? Because the obese mice, the leptin is deficient. That means they do not have a leptin. So without leptin, so they eat the food intake, the appetite will goes up a lot, right? Because leptin is the function of leptin is to suppress the appetite. So they have a very high appetite, eat a lot, and develop obesity and diabetes. So I think people were very excited about the discovery of leptin and thought, okay, leptin maybe can cure obesity and diabetes condition, but actually not. And they found that in the obese patient, in the obese people, the leptin level actually higher, not the lower. So the problem is they have a leptin resistance, in obese patient because although you have a high level leptin, but the leptin function is not normal. So so the different problems, so give us new challenges, right? And the other type of mutant mice that also related to leptin is called DBDB, diabetic obese mouse. And this mouse actually have a leptin receptor deficiency. So have a similar syndrome as a leptin deficiency, right? They also eat a lot and obesity and diabetic. But the cause is different. It's the leptin deficient. They have a probably normal or even high leptin levels, but leptin receptor deficient, so leptin cannot function in this mice. And they also, hyperphagic, and also develop type 2 diabetes, hyperinsulinemia. They have a high level of insulin, and hypoglycemia, high levels of blood glucose. And definitely the paralysis across circulation method were not going to work, right? Because the leptin receptor deficient. So you can give them leptin to cure them the problem. Okay, sorry. So now that's all information that I have for leptin. And now let's talk about the other important adipocyte-derived hormone, adiponectin. So adiponectin is also secreted from fat cells and has the two targets, but they all peripheral, right? So adiponectin does not have a central effect. So two targets, adiponectin are the liver and the skeletal muscle, right? So adiponectin can target liver and skeletal muscle, and the main function of adiponectin is also to stimulate fat oxidation, right? To stimulate burn more fat, energy expenditure is also help, right, with the obesity control or body weight obesity. So in terms of a regulation, it's also similar. Acute fasting, it can also decrease the adiponectin gene expression, which also makes sense, right? The adiponectin basically increase energy expenditure. So fasting, we want to save energy. So we also need to reduce adiponectin, and refeeding and similar like leptin, and also stimulate re-induce the adiponectin gene expression, and similar experiment they did insulin injection to the fasted animals can also increase adiponectin gene expression. And in the type 2 diabetes and insulin resistance, the plasma levels of adiponectin actually is decreased, right? So, which may be associated with some cost, of the type 2 diabetes. So, and this slide will just give you some idea in how leptin adiponectin worked to stimulate fat oxidation, right? Kind of mechanisms. Try to remember the AMPK. We talked about this kinase earlier, right? So AMPK, active protein kinase. So the main target inside of cells of both adiponectin and leptin is the AMPK. So both adiponectin, leptin can stimulate AMPK and stimulate fat oxidation that can reduce the triglyceride levels and fatty acids levels and decrease insulin resistance or increase insulin sensitivity. And this is just a general, the idea about the mechanism. And then we wanted to go a little bit more detailed about the AMPK pathways, how leptin stimulates this pathway and increase fat oxidation, right? So AMPK is a serum acerinin kinase and is a field sensing enzyme. That means the activity of AMPK can be regulated by the energy status, right? For example, if the ATP levels goes low in the cells, then AMPK can sense it and can be activated, right? So low ATP levels can stimulate AMPK activities. So that means tell cell, okay, we have a low ATP, so we need to make more like ATPs, right? So AMPK is activated and stimulates fat oxidation and produces more ATPs. So a lot of situations that, for example, starvation, definitely, right? We use a lot of ATP. We have low ATP, so then can stimulate AMPK, can increase the fat utilization in the cells and to allow the cell to make fatty acids for alternative energy fuels, right? So and also the other conditions, for example, glucose deprivation. So if we don't have enough glucose, then we need to use fat, right? So AMPK also need to be activated and to allow cell to be able to utilize fat as energy. And also hypoxia, right? Oxygen, lower oxygen condition that we also have a lower ATP production that will also activate the AMPK, right? And the consequences of the AMPK activation, that is the increased fat oxidation and glucose utilization. So AMPK is activated, can stimulate both glucose and fat oxidation, right? Okay, so this is a very important slide. You guys probably try to spend some time to remember, right? It's just the mechanism, the pathway, how leptin and adiponectin stimulate fat oxidation, right? And I can't remember, did I show this slide before? No? Okay, this is the first time. So you see that for example, this is the, let's say the muscle cells, right? And muscle cells have both leptin and adiponectin receptors. So leptin and adiponectin can bind to receptors, and then activate AMPK, right? We don't know details mechanism. But AMPK can be activated by high levels of leptin and adiponectin. And remember, the AMPK can regulate phosphorylation of ACC. Right? ACC is the enzyme that can catalyze the formation of marino-CoA. Right? So once the ACC is activated, and then will increase marino-CoA production, right? And marino-CoA, I think that we mentioned this before, right? Marino-CoA is very important, the substrate that can regulate the activity of CPT1, which is the enzymes on the mitochondrial membrane to regulate the transport of fatty acids from cytosol into the mitochondria for beta oxidation. Remember this process, right? So, and this is the whole, pathways, how AMPK involved in the regulation of fat oxidation by leptin and adiponectin, right? Again, first of all, regulates ACC and regulates marino-CoA production, right? And regulates CPT1. So again, AMPK is once it's activated, and that's the phosphorylate ACC, and ACC is once it's phosphorylated and the activity is decreased, right? So you have a decrease the production of marino-CoA. So marino-CoA levels goes low once AMPK is upregulated. And marino-CoA is the inhibitor of CPT1, right? So when you have a lower marino-CoA, you should have a higher CPT1 activity, right? Does that make sense? So increase CPT1 activities, and you have more fatty acids being fed into the mitochondria. So you have increased fat oxidation, right? And this, I guess, the last slide for the session. So, and the other important thing that I wanted to mention that in the skeletal muscle, remember, for the glucose uptake mechanism, insulin, of course, is one of the important mechanisms, right? It can stimulate GLUT4 translocation from inside of cells to the cell surface and to increase glucose uptake into the muscle. And the other important mechanism, like the muscle contraction, exercise can also increase the glucose uptake into the skeletal muscle. And so the mechanism for how muscle contraction stimulate glucose uptake is that also involve AMPK. So muscle contraction through some unknown signaling pathway that can also stimulate AMPK. And once AMPK is activated, and AMPK can regulate the translocation of GLUT4 from inside of cell to the cell surface, right? So AMPK increase can also increase GLUT4 translocation to the cell surface and increase uptake of the glucose, right? And this pathway is independent of insulin, right? The muscle contraction can directly stimulate AMPK and GLUT4 translocation, right? So two mechanisms, for glucose uptake and GLUT4 translocation in skeletal muscle. All right, so I think that's all for this session. So let's take a 10 minutes break. All right. Okay, let's move on. So let's start with the new uh, section, unit 3. I think I still have some metabolic fate of nutrients in response to intake, and then we're going to talk about the metabolic fuel utilization switch, right? Um, and I think in the last section that we mostly focusing on the different tissues, right? How different tissues utilize different metabolic fuels in fat fasting condition. And in this section that I think we more focusing on kind of timing, when um, uh, the more kind of slice precisely, to understand during the what time that we consider the fat condition and what kind of feed we use, and then fasting condition. And most important thing that I don't know if you probably already realize that the fuel switch, during the fat condition, we usually use more glucose as an energy fuel, right? And then during fasting condition, we use more fatty acids. So this kind of physiological switch is also kind of, the regulatory, right? So we're going to learn how our body to switch the fuel from using the glucose, carbohydrate in the fat condition to utilizing the fatty acids during the fasting condition. And so the first thing that we wanted to know is the whole metabolic condition that we can divide it a fat fasting condition, or we say the absorptive phase, right? Reference to the fat condition. So fat condition we also consider absorptive phase, and the post-absorptive phase or fasting conditions. So so absorptive phase or fat condition, we usually consider the 2 to 3 hours after meal, and during this period of time, we consider as fat condition or absorptive phase, right? So we know that after, for example, we eat breakfast, and after 4 hours, and we start to feel hungry, right? We know we need a second meal, right? We need to eat lunch. So from the breakfast time till like noon time, 2 to 3 hours, this time period is considered fat condition and absorptive phase. And post-absorptive phase of fasting condition. So, the lunchtime, we feel hungry, but if we don't eat anything, then start from 2 to 3 hours after meal and to the days of starvation. So if we don't eat anything, and the fasting condition can last whatever long, depend on when we start eat food, right? So can be short and can be long, it depend on, but so this is the first thing that we wanted to know, what happened during the absorptive phase, 2 to 3 hours after meal, and what happened to the post-absorptive phase, in terms of energy metabolism. And second thing is that how does the body switch metabolic fuels during the starvation, right? We know starvation, we utilize more fatty acids instead of glucose. So how our body does this. And then the third question that we are going to learn is how does the body know when to switch metabolic fuels, right? So this is, again, is a regulatory process, a mechanism. So mostly regulated by hormones and substrate enzyme activities and transporter activities, right? So, during the fat fasting, the major change in our body is the hormone levels, right? Insulin and catecholamine or glucagon levels change, and these hormones play a very important role in the regulating fuel utilization switch. So let's start with a standard meal. So a standard meal usually contains the 90 gram of carbohydrate and 30 gram protein and 20 gram of fat, right? So how carbohydrate is metabolized during the absorptive phase, the 2 to 3 hours after meal, right? During from 8 o'clock when start, when we eat breakfast and during the noontime. So how the carbohydrate is metabolized in the body. So so first of all, of course, we digest the carbohydrate in the diet and then absorb it, right? Remember this, we just try to kind of review, remember the SGLT1, this glucose transporters, right, responsible for the uptake of glucose from the small intestine to the enterocyte, right? And then enterocytes use small amount of glucose for energy. Remember the 10% of energy, right, in the small intestine is from the glucose. So glucose from the diet, and first of all, the use by the small intestine as energy, and then glucose then be transported into the portal vein to circulation by the GLUT2, right? Remember the transporters. And then will increase the high level of glucose in the circulation, right, after meal. So this will stimulate insulin release. And liver takes up 20% of dietary glucose by GLUT2. Right? GLUT2 is a major glucose transporter in the liver, which has a high PM for glucose. And again, the GLUT2, remember, is depend on the blood glucose level, right? Uptake activity. When blood glucose level is high and the uptake activity of GLUT2 is increased. So, and glucose is then rapidly converted to glucose 6-phosphate by glucokinase, right? It's in the liver, and most of the glucose taken up by the liver actually will be stored in the liver as a glycogen, right? In the fat condition or during this time absorptive phase. And the liver, of course, it can also utilize some glucose as energy, but usually glucose just goes partial oxidation in the liver, right? But this the partial oxidized glucose to the lactate in the liver, and this lactate will recycle and conserve glucose, skeleton carbon, and then glucose carbon skeleton for glucose production, and this glucose will be again saved as the energy in the form of glycogen in the liver. So in the absorptive phase or in fat conditions, the liver picks up glucose mostly is to store as a glycogen in the liver, right? Even some of the glucose be oxidized also, but just go the partial oxidation and save the carbon skeleton and convert it back to glucose as save as the glycogen. All right. So, and this slide just give us some idea, how much glucose from different sources of glucose contributed to glycogen in the liver in the fat condition. So 60 to 70% of the glucose in the glycogen is actually from the dietary glucose. So dietary glucose can directly be converted to glycogen, right? You know, dietary glucose first to converted glucose 6-phosphate and glucose 1-phosphate, and for the synthesis glycogen, right? So 60 to 70% of glucose in the glycogen actually comes from the dietary glucose. And the rest of 30 to 40% of glucose actually comes from the conversion right, from the lactate and amino acids, right? So in the diet, we also have amino acids, right? Protein. So amino acids in the liver can be converted to glycogen through this pathway, gluconeogenic pathway. Remember, we mentioned the gluconeogenic pathway not activated not only in the fasting but also in the fat condition in the liver, right? And this gluconeogenesis, the purpose of gluconeogenesis in the fat condition in the liver is to convert the lactate and amino acids to the glycogen, right? So, and also this pathway I just mentioned, you know, some of the glucose from the diet goes partial oxidation, right? But not complete oxidation, but just, you know, to the lactate, and lactate being reused as the precursor of gluconeogenesis, right? Come back, you know, converted to glycogen. So again, 30 to 40% of glucose in glycogen in fat condition is from the amino acids. And then the other 22% of glucose from the meal metabolized by the liver, and then the non-metabolized the glucose then released into the circulation, and then utilized by other tissues like brain. Utilize 20% of glucose, right? And the other tissues like 2 to 3%. And most of the glucose will be picked up by the muscle and the adipose tissue in an insulin-dependent manner, right? Remember, this is. And in the muscle, 25 to 50% of the circulating glucose that being picked up by the muscle, and 50 of them utilized for the complete oxidation for ATP production, and the other 50% of the glucose from the circulation is actually stored as a glycogen, right, in the muscle. And adipose tissue will also pick up some glucose, but for the two purposes, right? One, glucose can be utilized for the lipogenesis, right, for fatty acid synthesis, and the other purpose is to convert the glucose, convert glucose to glycerol 3-phosphate, right, through the glycolytic pathway, and for synthesis of triglyceride, right? So this all, I think the all information is about how glucose is metabolized during the absorptive phase, absorptive fat condition. And that's just the whole, the big picture gives you some ideas, you know, the 90% of carbohydrate glucose that we have in the meal that be metabolized, it goes to the different tissues distribution, right? 20 gram of glucose go to the liver for glycogen synthesis and also for partial glycolysis and for ATP production, and 20 to 40 gram the glucose goes to muscle, half for complete oxidation and another half for convert glycogen that's stored in the muscle, and 2 gram of the glucose go to the adipose tissue for triglyceride synthesis, right? And 15 20 grams of glucose go to the brain for complete oxidation, for energy production, right? So in brain, that particularly in the fat condition, glucose will be completely oxidized, right, for ATP. I think this slide we saw this before, right? This is so I'm not going to go through this slide again. I think we pretty much covered, right? This how glucose from the diet is distributed in different tissues and how they utilize and metabolism as as stored as glycogen in liver and muscle, and others oxidized for energy in multiple tissues. And proteins. So we have a 30 grams of proteins in the diet, and protein, digest and absorbed as amino acids and enter circulation, and 30% will be used to synthesize protein and store in the body, and the rest of the amino acids which just go to the catabolism. 13% will be metabolized by the in small intestine. We know that small intestine, the glutamine, glutamate, and aspartate are the very important energy fuels, right, for intestine use, and another 13% will be metabolized in the muscle. This particularly branching amino acids, right? Mainly metabolized in the muscle, and other 50% will be metabolized in the liver. So in the liver, 60% of amino acids in the liver can be converted to carbohydrate and stored as glycogen. We talked about this, remember, right? I think you guys just don't need to remember the percentage, but just give you kind of idea, how much amino acids or glucose metabolized in the different tissues or something like that. But you don't need to remember exact percentage, right? Okay, and this is just overall picture of the amino acids or protein metabolized in the absorptive phase, and 30 grams of amino acids that we can get from the diet, and 10 grams of the amino acid be used for synthesis proteins, and 12 grams go to the liver for the metabolites for urine synthesis, and glucose, converted to glucose stored in the form of a glycogen, or some of the amino acids can be oxidized as the energy in the liver. Remember, liver is amino acids, alpha ketoglutarate, right? Alpha keto acid, right? Is the most important energy fuse for the liver. The 4 grams of amino acid, particularly branching amino acids, will be metabolized in the skeletal muscle and kidney as well. And 4 grams of the amino acids, particularly glutamine, glutamate, aspartate, right, can be utilized in the small intestine as the energy. So this just tells us how amino acids metabolized in the absorptive phase. I guess we have a quiz, right? I'll just stop here.